Hi guys, this is Mrs. White coming to you with the lesson overview for section 10.2, the process of cell division. This is a little long, so I'm going to break it into two sections. So, what is a chromosome? So, a chromosome is that little X-shaped thing that helps DNA to become organized so that it can divide evenly and so that everything gets duplicated. Without being shaped into chromosomes, it would be like a spool of thread or a bunch of yarn that's just all tangled up. So there would be kinks and it would just be very difficult for it to duplicate very well. So the genetic information that goes from one generation to the next is carried by chromosomes. Each daughter cell gets its own copy of the genetic information, and every organism has a specific number of chromosomes. For humans, it's 23 pairs or 46 total chromosomes in each of our body cells. So prokaryotic chromosomes are a little bit different. Since prokaryotic cells lack a nuclei, their DNA is found just floating in the cytoplasm. Usually there's a single circular DNA molecule or a chromosome that contains the genetic information. In eukaryotic cells, chromosomes are located in the nucleus, and they are made up of something called chromatin. Chromatin is composed of DNA and histone proteins. So you can see the DNA double helix is getting smaller, and then it wraps itself around the histone proteins. The histone proteins then coil around each other and then supercoil into the chromosomes. As you can see, the center of the chromosome is called the centromere, and each chromosome that is duplicated is made out of two sister chromatids. The prokaryotic cell cycle is a regular pattern of growth, DNA replication, and cell division. Most prokaryotic cells replicate or copy their DNA once they hit a certain size. When they're finished replicating their DNA, they divide through a process known as binary fission. Binary fission is a form of asexual reproduction, which means that there is one parent and the daughter cells are identical. For example, bacteria reproduce by binary fission. In eukaryotic cells, the cell cycle consists of four phases: G1, S, G2, and M. Interphase is the time between cell divisions. It is a period of growth that consists of the G1, S, and G2 phases. The M phase is the period of cell division. In the G1 phase, cells increase in size and synthesize new proteins and organelles. So that's really our period of cell growth. The S phase, or synthesis phase, is where new DNA is synthesized when the chromosomes are replicated. So G1 is our cell growth, and S phase is our synthesis or DNA replication. The G2 phase is where we really get prepared for mitosis. Many of the organelles and molecules that are required for cell division are produced during the G2 phase. The M phase, or mitosis, is where we really get our division going on. In eukaryotic cells, division occurs in two stages: mitosis and cytokinesis. Mitosis is the division of the cell nucleus. Cytokinesis is the division of the cytoplasm. So mitosis, two nuclei. Cytokinesis, two daughter cells. So during prophase, the genetic information becomes visible and the nucleus condenses and the duplicated chromosomes become visible. Outside the nucleus, a spindle begins to form. During metaphase, the second stage, the centromeres of the duplicated chromosomes line up across the center of the cell. The spindle fibers connect to the centromere of each chromosome to the two poles of the spindle. During anaphase, the chromosomes separate and move along spindle fibers to opposite ends of the cell. And during telophase or telophase, the chromosomes, which are distinct and condensed, begin to spread out into a tangle of chromatin. So here's some important structures that we need to be familiar with. A chromatid is each strand of a duplicated chromosome. So 1/2 of a duplicated chromosome is a chromatid. The centromere is the central area where the chromatids are joined. It is also referred to as the waist of the chromosome. The centrioles are the tiny little structures located in the cytoplasm of the cells that help organize the spindle. And the spindle is a fan-like microtubule structure that helps separate the chromatids. So prophase. During prophase, the first phase of mitosis, the duplicated chromosomes condense and become visible. So prophase, you can see the chromosomes are becoming visible. The centrioles move to opposite ends and begin to form the spindle that will organize the chromosomes. And you can see that the nuclear envelope is starting to dissolve. So during metaphase, which is the second phase of mitosis, the centromeres of the duplicated chromosomes line up across the center of the cell. So metaphase is all about the middle. The spindle fibers connect the centromere of each chromosome to the poles of the spindle. During anaphase, you can think A, apart. The third phase of mitosis, the centromeres are pulled apart and the chromatids separate to become individual chromosomes. During telophase, the fourth and final phase of mitosis, the chromosomes spread out into a tangle of chromatin, and the nuclear envelope reforms. The spindles break apart, and a nucleolus becomes visible in each daughter nucleus. So the four phases are PMAT, or PMAT: prophase, metaphase, anaphase, and telophase. So just remember PMAT, and you can remember the 4 stages of mitosis. So the purpose of mitosis is to duplicate the nucleus. The purpose of cytokinesis is to separate the cytoplasm into 2 separate and distinct daughter cells. So cytokinesis is the division of the cytoplasm, and it's different in animal and plant cells. In animal cells, it pinches off to form 2 equal daughter cells. Each part contains its own nucleus and its own organelles. In plant cells, because they have this hard cell wall, they can't just pinch off. So what they do is they form a cell plate that divides the cell into 2 parts. The cell wall then forms in between the 2 membranes. So the stages of the cell cycle. Interphase, the longest stage of the cell cycle, is where the cell grows and replicates its DNA in centrioles. Then we move on to mitosis, our PMAT. Prophase, the chromatin condenses and you start seeing your chromosomes. Metaphase, the chromosomes line up in the middle. Anaphase, the sister chromatids move apart. And telophase, the nuclei separate and begin to form new nuclear envelopes. And then our final stage after mitosis is cytokinesis. The cytoplasm pinches in half, or in plants it forms a cell plate, so that you have 2 identical daughter cells with a complete identical set of duplicated chromosomes. Thank you for watching, and I'll be back with some new videos soon.